Hi folks, and welcome to the GTC conference. My name is Kartik. I'm from Vast Data and very excited to talk to you today about the advances in medical research that are going on using our technologies across the world. First thing to note is that classic NFS, just to give you a little more feel for what Vast is about, we are a storage platform that exposes ourselves through file and object protocols. And we are an all-flash system. I'll speak more about the architecture. But the traditional problem with file protocols, especially NFS, has always been that it's a bit too slow for the GPU workloads that we expect today. Classic NFS will give you about a couple of gigabytes a second as far as read throughput goes. While GPUs, you look at the DGX-A100, that gives you about 200 gigabytes a second line speed. Clearly, one might think that this is not an appropriate technology for the high-performance systems that we're working with today. The other problem that we typically run into in these kind of workloads is a question of scale. The architecture we have at Vast is a scale-out architecture, but one with some very unique characteristics. It has no cache coherency, and it has no east-west traffic anywhere. And it is truly the world's first shared everything scale-out system, which technically has no limitations as far as how large it can grow. We have customers who have 5, 10, 20, even 50 petabytes in a single namespace, and this is unique in the world of any kind of parallel file system or scale-out NAS, most of which are shared nothing systems, and we differ radically in this sense. How do we actually do this? Our architecture is very unique. We separate logic from persistence, and the logic layer, which also serves the file protocols, is completely stateless. This talks to the persistence layer, which uses a combination of Optane technologies from Intel and also QLC Flash, which allows us to keep the cost down extremely low in the system. Connecting these two is a high-speed RDMA network on which we run NVMe over Fabric. So every single container which is there on the stateless layer, on the file server layer or the logic layer, can see every single device in the entire high-performance cluster. This allows us to scale without limit, and when we hook a system up like this to a DGX-A100, we get spectacular kind of performance along with it. The economics, because we use QLC drives and very wide radius drives, as well as significantly superior data reduction technologies within our system, makes us affordable to the extent that we start to approach hard disk costs, even though we give the performance of large-scale all-flash parallel file systems. This has ended up in some exceptional performance for GPU-based systems, and our RDMA performance with the DGX-A100 has given us almost near line bandwidth to a single mount point for a DGX-A100. This is, of course, extensible beyond just the GPU world, and it works for all kinds of servers. But specifically to this talk, we'll be talking about how we leverage these technologies and how our customers leverage these technologies, specifically in the bioinformatics space. So let's start with looking at your classic genomics pipeline. So data from the sequencers typically goes into some kind of an engine which manages the workflow, and we go through the standard steps of genome alignment, variant calling, annotation, and other kinds of secondary or tertiary analysis for the genomes that are sequenced. One of the things I want to bring to your attention is about a year ago we collaborated with Intel to work on what they refer to as the Big Stack architecture, which is an implementation of the GATK framework, which is developed at the Broad Institute, which is pretty much one of the standard ways to be able to do sequence data analysis for genomics. We ran against the well-known human genome, which is the N812 H78 genome with input data with the human reference genome. And I show here both the workflow that we use as well as some of the results that we get. We were able to get on a 4-node Slurm cluster roughly one whole genome sequence in about 14 hours. But as you increase the number of whole genome sequence workflows, we were able to do about 40 of these in about 49 hours, or roughly 4.9 hours for sequencing an entire genome, which is extremely impressive. And the reason for this is because the VAST cluster is able to handle the tremendous amounts of I/O and bandwidth that are required when these jobs start up. And we used NFS using RDMA as a transport for this, and this really yielded superior results for us as we went on. We've also done the same kind of testing. This testing was done on DGX2s, and we use Clara Parabricks, which again is something which is owned by NVIDIA, to do genome sequencing. Very similar GATK pipeline. However, the difference here is that we start to leverage the GPUs, in this case the V100s on the system, to be able to do pretty much the same kind of whole genome sequencing that we did previously. Now, the results here are even more spectacular. We were able to get a single genome sequenced in a little under 40 minutes and do 2 whole genome sequences using 8 GPUs each in less than an hour. So that's about 30 minutes to sequence a genome. This is the kind of acceleration of science that we believe VAST enables for our customers. And this is one of the reasons why they are adopting our technologies in gusto. So another area of great interest recently is the field of nanopore sequencing. As you know, the typical short-read technologies which have been used for many years, are very effective in being able to sequence large genomes. But nanopore technologies are emerging as a strong alternative to what are referred to as long-read technologies. Now, what do I mean by these terms? A DNA strand has several base pairs or nucleotides which are paired together. And the idea behind sequencing is to figure out what is the order of these base pairs. And then by looking at different sections of this, we can look at different variants compared to reference genomes to understand better if someone has whatever, cancer or some sickle cell anemia or whatever the condition may be, giving the clinician direct and important insight into what conditions the patient may be actually suffering. Nanopore technologies use a very different technique from the traditional way to be able to do sequencing. They create nanopores, quite literally what it sounds like, a small hole in a membrane, and they apply voltages across these membranes. And then when a DNA strand is presented to this, there are some additional helper proteins which split the strand and feed one of the strands through this pore. Now, because there's an electrical potential across this membrane, current flows through this nanopore, and as the strand goes through it, each base causes a perturbation in the current, which is measurable. And depending on which base it is, adenine, thymine, guanine, or cytosine, the degree of electrical current change is different, and by using that, you can sequence what the initial DNA strand was. Advantage here is, instead of the 50 to 100 bases you'd get in a sequence chunk, or a read as it's called, you can have tens of thousands to millions of base pairs for each read, and hence the term long read. And this makes the whole task of sequence alignment a lot easier. And there's a whole bunch of new techniques that can be applied to be able to help the clinician understand better what's going on. So this base calling refers to looking at the electrical signals and being able to determine which DNA bases are present in that sequence. This turns out to be a non-trivial problem because you're doing pattern recognition, and modern state-of-the-art techniques use long short-term memory networks, or LSTM networks, or recorder neural networks, and combination of these to be able to do this kind of base calling. This is extremely GPU intensive. It's typically implemented in PyTorch, and there are standard ways to be able to do this. But this effort to do this kind of base calling is very, very intensive on the infrastructure. Once again, both on the storage as well as on the GPU complex. So we have many customers looking at these modern sequences as well to augment the science that they can do to advance the state of what humankind can do about illness and medicine and health. So of course, this is not the first area where we entered the bioinformatics space. We're a young company. We've been around for about the last 3 years or so. The National Institute of Health, for example, was one of our very early customers. And they ran much of their workload in their Helix cluster, also known as the BioWolf cluster, on parallel file system technologies primarily. They made an investment in us, and they took on about 8 petabytes of VAST storage, which replaced some of their parallel file system storage. Very HPC-like environment, over 5,000 client machines. And the HPC workloads primarily dealt with sequencing workflows. However, they also have started to do a lot of work in another very exciting field, which is cryo-electron microscopy. This is actually a field in structural biology, which allows you to see molecular structures at a level which is unprecedented by anything in the past, by using some exceptional techniques to be able to freeze samples in super cool water, in fact, to make sure the crystallization doesn't harm the tissue, and subject them to electron beams. There's been a Nobel Prize awarded for this kind of work, as most of you probably know. This cluster is now routinely running at a couple of 100 gigabytes a second. And the feedback from other customers as well who are running cryo-electron microscopy is unambiguous. That there are workflows and there are certain large samples, what they call multi-particle studies, which flat out will not run on many parallel file systems, and you need something like VAST to be able to run it. They're all, needless to say, absolutely thrilled with the work that we've done. This, of course, isn't the only kind of work that we do, genomics, genome sequencing, and cryo-electron microscopy. Standard image processing and deep learning techniques are also starting to come up extensively in the medical imaging field. For example, the MARTINOS Center, which is a joint collaboration of Harvard and MIT, uses AI for a variety of purposes. Now, some of the more interesting use cases include looking at lung function, especially in the days of COVID, when everybody's affected by this. This allows radiologists to look at successive X-rays of the same individual over a few days and use deep learning techniques to understand how much lung function has deteriorated. Why? Because this allows the attending physician to be able to quickly make a decision on when a person might need a ventilator or something like that. These are strong AI techniques, basically deep learning techniques, which use a lot of GPUs. In fact, MARTINOS was the first DGX-A100 customer in the world. They also run this on our technologies, and we're really, really proud and happy that we can help in the advance of science. Cryo-electron microscopy, of course, is now absolutely exploding in the structural biology arena. Dr. Sliz will be presenting immediately after this talk, is from Harvard Medical Systems, and he's done some absolutely groundbreaking work in the COVID virus for SARS-CoV-2, looking at some of the protein structures for the spike proteins for SARS, also using cryo-electron microscopy. That picture there gives you an idea of how big one of these machines are, but they are springing up all around the place like rabbits. We see them all over the place. Most large research facilities have 1, 2, 4, 8 of these, even more. And increasingly, this is becoming mainstream as far as analysis goes. That's exactly the reason why VAST turns out to be a very, very popular platform for people doing structural biology work as well. Of course, I should mention that this is not the only thing that actually VAST is good for. We call ourselves universal storage. Our architecture allows us to scale to both transactional as well as analytical workloads. So, of course, in life sciences has been one of the anchors for us from a business standpoint, but that's not the only vertical we're known in. We do very well in finance and content, high-performance computing. Many secondary use cases as backup targets, as well as clearly big data and AI is very, very hot on our list. And we do a bunch of work in enterprise use cases as well. VMware, CSI environments with Kubernetes, all of that is very much in our wheelhouse. I hope you'll enjoy the talk right after this by Dr. Sliz. I think the work he's doing is absolutely groundbreaking. Looking forward to more advances in this field from using these really exciting technologies. With that, have a fantastic day, and we will meet again at the next conference. Thank you.